All right, back to chapters 24 and 25, which I do believe I just said is boring. So this was where we're going to get into talking about a little bit of environmental and applied microbiology. A lot of this stuff is going to be getting into more of how microbes are affected by and affect our environment. So one of the terms we talked about in this chapter 24 was biome. A biome is a climatic region. So literally, let's talk about the climate. Okay, a terrestrial realm of the biosphere. So that's where anything that can live would be living. That's the biosphere. It's characterized by certain weather, flora, and fauna, right? So this is like the savanna and things like that, the rainforest. We learned about this in middle school, right? So that term for that is a biome. We also talked in chapter 24 about the food chain, primary producers. These are organisms that make their own energy. And, basically get their energy and their, carbon sources and stuff. They get that from the environment. They don't rely on other organisms to provide that to them. So they use a lot of inorganic resources or use sunlight for energy, all that stuff. These guys are the basis of the food chain. They are the foundation of the food chain always. It's always the primary producers, not primary consumers, primary producers, okay? They're producing their own stuff. Primary, they're the beginning, they're the start, of the A number one. Everything from there on in a food chain relies on the primary producers to some degree, some point. Even when we're talking about the saprobes that break down dead materials, they still are getting their energy from stuff that was made by primary producers originally. So everything relies on a primary producer. So these are going to be our photoautotrophs, if you remember those terms from earlier. That's one of the examples I'm going to use. So that's the food chain. We talk a little bit about like the carbon cycle and everything like that in chapter 24. I do recommend kind of watching through or reading through at least the lecture notes for that one. But carbon cycle, basically the watered breakdown of that situation is that there's carbon dioxide in our atmosphere from combustion, from exhalation from aerobic respiration. We have that as a waste byproduct from the Krebs cycle, remember? And essentially all that carbon dioxide builds up in the atmosphere, and photosynthesis can utilize sunlight to create energy that can help drive fixation of carbon dioxide from the atmosphere into usable glucose or other like carbohydrate type compounds. So that's how photosynthesis plays a role in carbon cycle. I think that'll probably be the more important one. Remember, microbes are the number one photosynthesizers on the planet. That's going to include our algae as well as like our cyanobacteria. Which remember, algae are going to be using chloroplasts for photosynthesis. That's the organelle that performs photosynthesis. Whereas cyanobacteria, they don't have organelles. So that's going to be happening on the inside of the inner membrane of those bacterial cells. All right, and then the importance of nitrogen in soil. Plants need to make DNA and protein as well, and those things are pretty heavy in nitrogen. Depletion of nitrogen in the soil is a huge problem right now. That's due to global warming and climate change. This is an actual real serious problem and may lead to starvation in certain parts of the world, just because they aren't able to grow foodstuffs as well, vegetables and fruits, as well as they used to be able to, because the nitrogen in the soil is being depleted because the bacteria aren't able to grow there as well due to the climate changes, right? So soil biology is extremely important as far as maintaining nitrogen in the soil goes. And if we are, like we have said just previously, just a second ago in our talk, we mentioned, listen, what I thought I had. Okay, we mentioned primary producers and how everything below them relies on them for, food and energy and everything like that. And now I'm telling you that they're being depleted. Those primary producers are being depleted from our planet because of climate change. This should be a concern to you. And it is to a lot of people that know about this in the world. So definitely be aware of that. All right. Climate change is real. So we have potable water. That's a term that refers to water that's free of pathogens. It does not have to be clear. It does not have to taste nice. If you have put iodine tablets into your water and it tastes of iodine and it's all orangey from the iodine tablets, and you have to drink that, but there's no pathogens in it, then that is potable water. It is pronounced potable, not potable. Potable from potare, it's the Latin for basically drinkable water. Okay. Water filtration and wastewater treatment. So whenever water is coming into your house for you to actually consume, that's going to include a bunch of filtration steps, moving large matter like rocks and things like that out of the water, and then actually like filtering smaller and smaller, and then exposing things to ozone or chlorination to remove any potential pathogens. Then we have release of wastewater into the environment. Yes, that's what happens. Okay, so if your waste, literally like your sewage, is sent to a wastewater treatment plant, they are going to remove as much of the sewage aspect of it as they can. So they'll use even bacteria to break down sludge, and that is just the solid portion of the sewage. They'll filter out the liquid portion, and then usually incinerate or prepare for use for, whether it's fertilizer or something like that, the solid part of the waste, right? So like often incinerating that. And then we have the liquid portion, and we're going to have to go through several cycles of exposure to air, as well as bubbling through air, as well as treatment with like chlorine and filtration and all of that stuff, right? And the whole idea with this is that we can get enough of the actual bad bacteria out of there that we can safely release it back into the environment, instead of letting just waste go out into the environment, which can lead to a lot of illness, and does lead to a lot of illness in underdeveloped countries. When I say about bubbling air through, can any of you guys think of reasons why you might want to bubble air through sewage? The reason we would want to bubble air, just air, like what we breathe through, is because air has oxygen in it. And a lot of the bacteria that are found in sewage are anaerobic bacteria. So they don't have the ability to defend against the toxic products that are made from oxygen, the way that aerobic bacteria do, the ones that undergo aerobic respiration that we had talked about in chapter 8. So I think that is a good way to sort of like bring that stuff full circle. We monitor the safety of the wastewater that we are releasing back into the environment by monitoring the coliform count. So these are indicators of presence of pathogens. So what kind of pathogens might we be concerned about? Like cholera, or E. coli 0157H7, which is the S-tech E. coli, the Shiga toxin producing E. coli. We'll talk about that a little when we get into diseases in unit 4 towards the end of the semester. Salmonella, all sorts of these bacteria that could be transmitted as pathogens to people and make them very sick. There are other bacteria that we can look for, that if those less harmful bacteria, so just normal E. coli or other coliforms, enteric bacteria, normal enteric bacteria, if we find those in the sample, then we could potentially find the really bad pathogens in the sample. So those are our indicator bacteria. Those are coliforms. And what we basically do is we will, dilute out the sample of the water and then count how many coliforms there are. And we have a limit that is acceptable, whether it is in drinking water, whether it is in gray water or swimming water, or even wastewater that's being released back into the environment, which would be the higher level compared to all of those. But yeah, we can only have so many bacteria present per milliliter of water. Yeah, obviously, I feel like that makes sense, right? So what else are microbes good for, other than like they cause disease? We know that, but do they do anything good for us? Well, obviously they do, right? So we are going to be talking a little bit about fermentation to make bread or beer or wine, or pickling foods, yogurt, cheese, other dairy products. All of these things involve the use of microbes to our advantage. A lot of the times fermentation is the reason for all of that being produced. The gas leading to the little bubbles of air inside of Swiss cheese. That's a gas product from fermentation. Lactic acid production causing an acidic quality to pickling. Production of alcohol as a byproduct of fermentation in the production of beer and wine making. Often that has to do with both yeast and bacteria. And same thing with bread, right? So yeah, so I think that's pretty cool stuff as far as what that's good for. To keep the bad guys out of our food, we have a lot of different tests that we can do for food safety and things to prevent those guys from growing. Cold and freezing, it doesn't kill anything, it'll just stop them from growing. So when you get them back out of the cold, again, later, thaw them out, they're just going to start growing again. So keep that in mind. Cold isn't going to kill anything. You have to cook things to certain temperatures in order to denature the proteins that allow those organisms to function. And that's what cooking does. It heats up the proteins and denatures them so they cannot function anymore. And that is what kills the bacteria. And we'll talk about that in the next, in chapter 11, when we get into that. I know you guys have a worry about the midterm first, and that is fine. So we'll deal with that. But yeah, that's going to lead us pretty well into chapter 11, starting off with unit 3. Maintaining basically a clean environment, how we kill microbes, how we control microbial growth and everything like that. So cold only puts things into stasis, it doesn't kill them. And that's pretty much that for the first half of the course up to the midterm. And we will have our review on Zoom, and you guys are welcome to join into that, and I will give you guys the link. The link is already provided, sorry, in the website, where it says join Zoom. That's where any of our online stuff is going to be happening if we do it over Zoom. Same link every single time. Other than that, I'll give you guys the time and date and everything like that. Yeah, when we get there. So look out for that on Canvas. And I am obviously recording this well beforehand, so I don't know what day that's going to be yet. Sorry. But you'll get an announcement. Don't worry, and I'll see you guys next time.